Word. Okay. Hey, everybody. Welcome to the second exam review. Just like the first one, this is going to open on a Saturday at noon. You have a full 24 hours to do it, so the exam will close Sunday the following day. Format is exactly the same as exam one. Laura's going to upload the exam Word doc. You could download that right directly into the document and then upload it before that deadline. If you have any questions or time conflicts, accommodations, please take that up with Professor Francis. If you have a question like while you're taking the exam, Laura told us that she predominantly wanted to handle the questions. So email Laura if you have any questions, but if you want to email one of us as well, that's fine. Just make sure you tag Dr. Francis too. Okay. How do you want to, so, okay, I'll go over this, and I know, Rachel, you like the RTK. Pathway. Yeah, so I'll just get it started with this.
Okay.
So main ideas of cell signaling. Cells are always receiving signals for a bunch of different things. So it's constantly receiving survival signals, or when it's time to replicate and grow, it might start receiving growth signals. So the signals just tell the cell what to do. If the cell stops receiving signals, it will most likely go into programmed cell death. This is why harvesting cells or growing cells in vitro, so outside of the body, is pretty difficult, because it's very hard to replicate all of the molecular signals that are happening inside the body. Okay. Oh, and something I just want to point out, if you guys have any questions about anything, feel free to interrupt me or drop it in the chat. I'll try to be paying attention to that. But if at any point you have any questions, like I said, feel free to just interrupt me. Okay, so receptors, they're your extracellular ones. These are the ones that we're mostly going to be talking about with the ones outside of the cell, but there are also intracellular ones. There's some extracellular signals that can actually be absorbed into the cell by endocytosis. The main receptors that we're probably going to be talking about is the RTK, the receptor tyrosine kinase, that comes up in a couple different pathways. Okay, signal amplification and crosstalk. So the general idea, or I guess the main idea of these, is notice here, I'll talk about signal amplification at first. So you have a ligand that connects to the receptor. The receptor talks to the downstream protein, that talks to one downstream protein, all the way until the full transaction is done. The idea of signal amplification is that instead of one protein activating only one protein, activating another protein, whatever, it amplifies the signal. So instead of the receptor activating one, it could activate multiple proteins, and those multiple activate other multiple proteins. It's more efficient to get the signal out, because of course, it amplifies the signal. So if the cell wants something done in a short amount of time, and it wants it done abundantly, signal amplification is a really great way to do that. Another way is crosstalk. So some of the pathways that we're going to talk about, like the survival signal, the RTK, RAS, MAPK pathway, all of those, they're pretty linear. We're going to be talking about them in a pretty linear way, but just know that in real life, crosstalk is actually happening all the time. So some signals in, let's say, the RTK MAP pathway can communicate with some other signal in the G-coupled receptors, which can also communicate or disable something in their survival signaling. So these signal transduction pathways are very rarely just very linear. They kind of intertwine with other signals, as you can see in this diagram. Wait, am I blocking this diagram by any chance?
Very good.
Okay, sweet. Yeah, as you can see with this diagram, all of the signals, they're not very just linear. They're kind of talking to a bunch of other proteins here. So that's the general idea of these two. Do you guys have any questions about anything so far?
Nope.
Okay, sweet. Okay, Rachel, I know this is your favorite pathway, so do you want to take this one?
I just remembered from when I was in Laura's class, for some reason, this pathway just was imprinted in my brain. So, okay. So RTKs, or receptor tyrosine kinases, are a family of receptors that are used for survival, cell division, cell differentiation, and insulin signaling. These are the main ones they're used for. They're used for a few other things, but these are the main four things they're used for in the cell that you should know. And then just a little recap, a kinase is an enzyme that catalyzes phosphorylation. So this is just the reaction of phosphorylation with the kinase. You can go to the next slide. Okay. So with RTKs, this is their general structure. They basically start.
Sorry,
My
bad.
There you go. They basically start as two RTKs, and then the receptor will come in, or not the receptor, the substrate will come into the receptor domain, and then cause the two like RTKs to autophosphorylate themselves. It's a transmembrane receptor, so that just means it goes through the membrane, and it has this tyrosine kinase domain, which is where those like little black thingies are. So these are the steps. The substrate binds. In this case, this is a dimer, I think. Yeah. And it binds to the receptor domain, causes them to dimerize, and that dimerization stimulates the kinase activity, which causes this cross phosphorylation. So they're allowed to autophosphorylate themselves to create binding sites for future pathway proteins. Any protein that is able to bind to the phosphate has a phosphotyrosine binding site, which is the SH2 domain. So if you remember that from the RTK pathway, that's what the SH2 domain is what binds to this. So like a protein that can bind to an RTK has to have an SH2 domain. So in the RTK pathway, I believe it's GRAB2. Yeah, GRAB2 binds to the RTK because it has a SH2 domain. So this is just a flow diagram. Two other TAs that taught in the fall made this, but so the RTKs are used in two pathways that we talk about, which is the RAS signaling, which is the RTK MAPK pathway we'll talk about in a second, which leads to this MAPK cascade. And then they're also used in the survival signaling with AKT, if you remember, and BAD and BLC2, which trigger for a cell not to go through with apoptosis. So this is what the two RTKs are used. So yeah, you're good. Yeah, you're good. So for the RAS signaling or the RAS MAPK pathway, we start with that dimerization. The substrate binds to the domain on the outside of the cell, and then all of this occurs on the inside of the cell. So the RTKs autophosphorylate themselves. GRAB2, which has a SH2 domain, can bind to the phosphate on the RTK. GRAB2 then recruits what's called SOS, which is also a RAS-GEF. And RAS-GEF can bind to GRAB2 through the SH3 domain that it has, which allows SOS to bind to it. Without this SH3 domain, SOS wouldn't be able to bind to GRAB2. And SOS has a PXXP motif, which is just a little area on the that side of the protein that binds to the SH3 domain. So the PXXP motif allows SOS to bind to GRAB2 through GRAB2's SH3 domain. I know it's kind of a lot, but this little diagram at the bottom kind of shows you where the domains are on GRAB2. See how SH2 is on the side that binds to the RTK and SH3 is on the side that binds to SOS. And there's nothing that indicates that SOS has a PXXP motif, but it's there on that side that's connected to GRAB2. And SOS is a RAS-GEF, which means it switches GDP for GTP. And so what it does is it does this by activating RAS. So kind of like how RAN-GEF did this in nuclear import and export. RAS-GEF activates the RAS protein. So GTP diffuses it, which allows the signal to be transmitted further. This is just a little side note. There is a RAS gap that can stop the transmission, and it does this by inducing GTP hydrolysis. And then one other thing I want to mention is the RAS protein that RAS-GEF is activating has this little, see that little like black line? That's just a lipid anchor. So it never leaves the membrane. It will only move like side to side. It won't ever leave it because of that anchor. That was kind of a lot. But do you guys have any questions on the first part of the MAPK pathway? I'm going to take that as no. Cool. All right. So this is the second part of the RAS-MAPK pathway. MAPK stands for mitogen activated protein kinase. You don't really need to know that, but that's what it stands for. So if you remember from the last slide, we now have activated RAS with GTP instead of GDP, thanks to SOS. It's now activated, and it can activate MAP kinase, or MAP K3. And then this kind of cascade of events occurs. So MAP K3 can then phosphorylate MAP K2, which can then phosphorylate MAP K. So it kind of just goes down this chart of how they activate each other, and they do this by phosphorylation. And then once MAP K is activated, it can then go on and phosphorylate whatever proteins it needs to in whatever the cell needs. So if it needs changes in protein activity, MAPK will phosphorylate these proteins that will change their activity, probably make them active. Phosphorylation can also make proteins inactive, but in this case, we're just going to say it actually doesn't specify. It could be inactivating or activating these proteins. It just changes the protein activity. And then if the cell needs a change in gene expression, then MAPK can phosphorylate these transcription factors, and that will cause a change in gene expression, whether it's to inhibit or activate it. And then the reason these ATPs are here is because kinases always use ATP as a source of phosphate when phosphorylating something. So because these are all kinases, they're using this ATP and taking that one phosphate from ATP to turn it into ADP. So we're going from 3 phosphates to 2. And that's what phosphorylates these kinases. And then like the proteins or transcription factors, whatever it's phosphorylating, it uses ATP if it's a kinase. So just remember that it goes from K3 to K2 to K1. So it goes down in numbers to get your overall result in the RTK MAPK pathway.
All right.
Do you want to do this one, Sebastian?
Sure. This is RTK. Okay, so similar to the pathway that Rachel just talked about, we're still dealing with the RTK receptors. So a signal is going to come in. It's going to dimerize these 2 receptor tyrosine kinases that allows them to autophosphorylate themselves. So once they're active and phosphorylated, in the signal, or excuse me, in the survival signal pathway, it's going to recruit this big protein here called the PI3 kinase. PI3 kinase, notice that it's binding to the receptor tyrosine kinase, so it must have an SH2 domain because obviously it's binding to the kinase. So what PI3 does is it activates this inositol phospholipid called PIP2. It's going to activate PIP2 to make it PIP3. PIP2 already has these 2 pre-existing phosphate groups. All that PI3 does is it just adds the 3rd phosphate group to PIP2, which makes it PIP3. There's another protein, P10, which can remove the phosphate from PIP3 to inactivate it again, but I don't think that's extremely relevant, but just so you know. Okay, so now that you have PIP3, now that the inositol protein is phosphorylated 3 times, it's able to bind to this big protein called AKT. So AKT is going to find the 3rd phosphorylated PIP3, and it's going to connect to it. However, it's not active yet. Before AKT can become active, it first needs to obviously bind to a PIP3, but then it also needs to be activated by 2 different protein kinases. There's protein kinase 1, which here, notice it's also connected to another PIP3. This is a different one. This is a different PIP3 than the one bound to the AKT. But this protein kinase 1 is going to phosphorylate AKT. Another protein kinase is going to, again, phosphorylate AKT. And once AKT is doubly phosphorylated, then it's able to transduce its signal. This is just like the word explanation, but I'll explain it in, where'd it go? Oh, okay. I'll explain it in the diagram. Okay, so there's 2 scenarios. The first scenario is where BCL2 is bound to BAD. So this complex just exists like all the time. If BCL2 is bound to BAD and it cannot release itself from BAD, the cell is most likely going to go into apoptosis. But because cells are, like I mentioned before, are constantly receiving survival signals, it's activated the survival signal pathway. So in the second scenario where PIP2 got phosphorylated into PIP3, recruited AKT, AKT got doubly phosphorylated. Once AKT gets phosphorylated twice, it's able to release itself. It's active and it's able to dissociate from PIP3. Once it dissociates from PIP3, it's going to find this BCL2 bad complex. It's going to phosphorylate bad, which changes its conformation, no longer puts it in a place where it has an affinity to BCL2. So in other words, it increases the KD for BCL2. Once BCL2 is relinquished from bad, it's able to go, I believe, to like the mitochondria or something and inhibit apoptosis. Okay, so any questions about this pathway or any of these steps? Okay. So
I can do Jack
okay,
stat.
sure.
Do you want to do, and then do you want to do GPCR like we did with
Yeah.
the?
Oh, true. Okay.
Yeah, because I think that's the next one. All right, so Jack stat's a short one. It's it's not many steps. But anyways, so Jacks, we're no longer talking about RTKs because Jacks are different enzymes. They're just subunits on top of the receptors that have enzymatic activities. And in this scenario, we're talking about prolactin receptors, which are used to make milk in breast tissue. So basically, these two JAK subunits are located on the receptor in the membrane. So they're once again transmembrane receptors, but everything else is occurring on the inside. So basically, what happens is prolactin comes in and binds to the receptor, in a similar way that the RTKs dimerize. These receptors come together, and then the JAK. Wait, I'm just reading what they wrote. Yeah. So the JAK subunits can then cross phosphorylate each other, like they are doing where Sebastian's cursor is showing. They do this once the receptors come together and allow them to activate themselves with phosphorylation. Once they cross phosphorylate themselves, they then phosphorylate the receptor itself. So if you see in the next step where Sebastian's cursor is, these two phosphates are added by the JAK subunits, and these create binding sites for STATs, which are I don't actually know if they're enzymes. I think they're just proteins. They're just proteins. So they come in and bind to this phosphate with an SH2 domain, similar to how GRAB2 binds to the phosphate in the RTK MAPK pathway. So they bind to the phosphate, and once they bind, then the JAKs phosphorylate the STATs themselves. So now you have not two phosphates on them, but you have the STATs binding to one phosphate, and then they have a phosphate on them. Once they're phosphorylated by the JAKs, they then break off and bind to each other at the spots where they were phosphorylated. And this makes them active. So now they're a dimer and they are active and can then enter the nucleus through the nuclear pore. And then their job is basically to bind to transcription factors and change or alter or enhance or whatever you want the transcription of milk protein genes in this specific scenario for prolactin. And they do this by binding to the transcription factors, I guess the transcription factors bind to the STAT dimer, but either way, it alters transcription. Yeah, that's it. That's the JAK STAT pathway. It's pretty quick, pretty short.
Okay, the G protein coupled receptors. I mentioned last time that this is the most common kind of receptor in eukaryotic cells. So this pathway is involved in a bunch of different things. If you read primary literature, you'll see these pathways come up several times. So the way it works is you have your G protein coupled receptor right here. It's also a transmembrane protein. It's going to receive some kind of ligand, some kind of signaling molecule. And when that happens, the G protein coupled receptor, this giant green thing, is going to activate its GEF analog, which means that it's essentially going to function as a GEF. It's going to, before that, this giant green molecule, the G protein coupled receptor, is coupled with these three subunits, G alpha and G beta, G gamma. Okay, so once the signal molecule comes in, it activates the GEF analog. It exchanges GDP for GTP, and that activates G alpha. once G alpha is activated, so once it's holding on to GTP, it's going to dissociate from the G protein as well as the beta and gamma subunits. Sorry, once it dissociates, G alpha is able to go all around the cell, do whatever it needs to. Oh wait, I think it is, yeah, sorry, never mind. It's going to be anchored onto the phospholipid bilayer. So it can't just like float around, diffuse everywhere. It's still anchored, but it is going to move around a great deal and start activating whatever it needs to activate. So after a while, once this pathway is satisfied or once it's activated or deactivated or whatever it needs to, another protein comes along, GRS, which has a gap analog. So it's essentially going to function as a gap. It's going to hydrolyze the GTP into GDP, which leaves G alpha connected to GDP. And once G alpha is connected to GDP, this complex is going to reassemble again and the pathway is going to be turned off. So G alpha is again going to be connected to the G protein coupled receptor as well as the beta and gamma subunits. And that allows for this process to just happen over and over again. Okay, so do you guys have any questions about any of the past two pathways?
I actually have a question about the survival signaling pathway.
Sure.
When is that, like, what's an example of where that would be used?
So this one?
Yeah.
Actually, always. Cells are always receiving survival signals. In the event, so this process with the bad complex being dissociated and BCL2 clogging up mitochondrial holes, I know that it has some role in the mitochondria. I remember reading that it blocks mitochondrial holes. I don't know why these holes are forming, to be completely honest. I just know that they play some role in the mitochondria by like clogging these holes. And like I mentioned before, cells always need to be receiving survival signals. In the event that a cell stops receiving survival signals, so when the survival signal is no longer present, these guys can't dimerize, which obviously like the whole downstream effect can't happen. And your bad BCL2 complex cannot dissociate, which I guess allows these mysterious mitochondrial holes to expand. And of course, if you have no mitochondria, you have no energy inside your cell. So that ultimately leads to apoptosis. So this pathway specifically is relevant always. It's happening all the time in cells that like want to be alive. If a cell has like serious damage or the body just doesn't need it anymore, then of course it'll probably go into programmed cell death. But for cells that really do want to stay alive, that need to stay alive, this pathway is happening all the time. So does that answer your question?
Yeah, and then could you also explain that type of thing for the RTK, the RAS, that one?
This one?
No, the RTK and the
uh,
map, yeah.
This one, yeah, so this one's more specific to other things. So the outcome of this one, and correct me if I'm wrong, Rachel, because I know this is like your thing. But this one, it can alter like protein activity. So as you can see here, it's like changing a protein shape. And when you change the shape, you change the function. So maybe this pathway is like activating this protein to activate other downstream proteins, or maybe it's deactivating it so that it doesn't transmit that signal. It's activating transcription factors for gene expression, as you could see here. Yeah, Rachel, is there anything you want to add to that?
No, that's pretty much it.
Okay. Yeah, so this pathway, unlike the survival signaling, which is happening all the time, this pathway is to accomplish like a specific goal. So like I said, alter protein activity or to get some transcription factors going for a specific gene. If I had to guess, I'd say the survival pathway is much more active, or it's active more often than this one is, because this pathway, it's for like a specific goal, for like one specific outcome. But the survival signal is for the entirety of the cell's lifetime. So does that answer your question?
Yeah. Perfect.
Okay,
Thank you.
no problem.
If I could ask a quick question.
Sure.
Just with the JAK-STAT pathway, my question is, so there's JAK1 and JAK2. I'm just wondering, is there any difference between JAK1 and JAK2? Because they kind of are portrayed as if they're doing the same thing.
I don't believe there's any difference. They both have the same function, and they're both just cross-phosphorylate each other. So I think it's okay to say that they're the same. I mean, I think they're just subunits, like different subunits, but with the same function. Like one is labeled JAK1, one's labeled JAK2. I think you could reverse the names too. They're basically like twins, kind of.
Right.
All right, great. Thanks.
Yeah. Okay. So the cell cycle. I'm going to go through all of these steps in more detail in like separate slides, so I'm not going to spend too much time here. The only step that I am going to talk about is G0. G0 is essentially your non-dividing state. This is where cells just be cells and they just do their thing. Some cells stay indefinitely in G0, so for the entire lifetime. So like neurons, for instance, that don't replicate, red blood cells don't divide. So there are some cells that stay indefinitely in G0. Some cells can actually enter the G1 phase. So like maybe they're about to divide, but even sometimes they can enter G1 and they could realize like maybe it doesn't have enough nutrients or maybe it's not large enough. It can actually go back into G0. There is a point in G1 called the restriction point, which again, I'll talk about later. That's the point of no return, where once a cell passes that restriction point, the point of no return, it can't go back to G0. The only outcome is you either complete the cell cycle or you go into programmed cell death. Obviously, we're mainly going to be talking about the cells that do engage in the cell cycle, considering that this is the cell cycle chapter. So how this happens is, or how the cell cycle is essentially regulated is you have these CDK molecules, cyclin-dependent kinases. They're called the master cell cycle regulators because they're the ones that are essentially activating, deactivating, and getting the whole cell cycle going. So some ways that you could regulate them is through cyclin levels. So hopefully this is review for you, but CDK alone really can't do anything. CDK is dependent on a cyclin. If CDK is not bound to cyclin, again, CDK really has no purpose. It has nothing to do. So one way that you could regulate CDK is by regulating cyclin levels. Another way that you can regulate it is, so like I mentioned, CDK must be bound to cyclin for it to do anything. But in addition to being bound to cyclin, the CDK and cyclin complex must also be activated with a different protein. So CDK alone doesn't do anything. CDK and cyclin together in a complex still actually doesn't do anything, not until it is phosphorylated. So that's another way that you could regulate this. And lastly, another way is through CDKIs, which are CDK inhibitors, which of course I'll talk about in more detail later. Yes, so CDKs need both the cyclin and the activating phosphate for CDKs to be completely active. And like I mentioned, these are the master cell cycle regulators. So if at some point the cell reaches like a checkpoint or something where it wants to make sure, is there DNA damage? Is the cell large enough? What it's essentially going to do, it's going to deploy something that can inhibit CDK, being that CDK is the master cell regulators. When the cell wants to put a brake on the cell cycle, it's almost always going to target the CDK. Okay, any questions about that? Okay, so cyclins, like I mentioned, they are coupled with CDK. These are a little more specific. There are different kinds of CDKs. I don't think you need to know like too much about them. But cyclins they do come in different forms, I guess. There's CDK1, CDKE, CDK. Oh, sorry. I take that back. Cyclin. Cyclin comes in a little more forms. And it actually depends. The cell cycle needs the CDK bound to a specific cyclin. So as you can see here in this diagram, all throughout G1, CDK must be bound to cyclin D1 and maybe a little bit of cyclin E. But once you go into S phase, CDK now needs to be bound to maybe a little bit of cyclin E, but it predominantly needs to be bound to cyclin A. That also goes for G2M phase. Once you get into G2M phase, CDK needs to be bound to cyclin B. So as you can imagine, these cyclin levels are constantly fluctuating because CDK needs to be bound to a specific cyclin for it to accomplish whatever it needs to accomplish, like whatever proteins need to be activated or deactivated for that particular cell phase. There's not a whole lot of ways to regulate cyclin other than transcription levels. and through proteolysis. So if the cell doesn't want any more cyclin D1 because it's now transitioning into S phase, it's going to either downregulate, that's the word. It's going to downregulate transcription for cyclin D1. If there's a lot of cyclin D1 present, it's going to undergo ubiquitin-mediated proteolysis. Okay, any questions about cyclin? Okay, so here's one example of how the cell cycle can arrest it. Let's say that the cell is doing its thing in G1, checking for everything, and it notices that there's DNA damage, or there's a mutation, or whatever it may be. P53 is going to be activated. P53, it's a transcription factor, oftentimes called like the tumor suppressor. And what it does is it activates the transcription for P21. P21 works as a CDKI. It's a CDK inhibitor. And again, when you inhibit CDK, you put a halt on the cell cycle. Because CDK is extremely important in progressing through the cell cycle, if you inhibit it, you inhibit the cell cycle. So P53 is super important to prevent things like cancer, or to make sure that the cell can properly fix whatever DNA damage may have occurred without the cell continuing through the cell cycle. If the cell is paused because it's repairing DNA damage, and the DNA damage is like irreversible, or there's too much damage, it's most likely just going to go into programmed cell death. Okay, G1. So cells are usually in G0, they're just doing their thing. And then when it's time for them to replicate, they're going to enter G1. So in G1, they're going to be receiving a bunch of signals. So like those signals that we talked about in the previous lesson, it has to be receiving a bunch of different growth signals, which of course has like their own pathway. So to be in G1, like I said, the cell needs to receive those growth factor signals. It's going to start checking for DNA damage. If it detects DNA damage, it's going to deploy that P53 that I just talked about. It's also going to start producing G1 cyclin, or cyclin D, which is the cyclin that corresponds to the G1 phase. It's checking for size, that it has the right size to divide, it has enough nutrients or energy. This is also the phase where the pre-RC, or the pre-replication process, is formed. Okay, so here's essentially what the pre-RC is. The pre-RC, like I mentioned, is formed in the G1 phase. It cannot form, so it forms when CDK levels are low. And that's because you don't want CDK activity to be high, because then this CDK is essentially what's going to fire the pre-RC to get it into the pre-IC. But if there's plenty of activated CDK running around the cell everywhere, it's going to, it might actually accidentally prematurely activate the pre-RC and get it started into the pre-IC, which of course, if the cell isn't large enough, or it doesn't have enough nutrients or something, that could be pretty consequential to the cell. So the pre-RC is actually inhibited by CDK levels. Okay, so what the pre-RC is on the DNA, there's the spot called the origin of replication. That's where, as you might have guessed, a replication is going to originate. On the origin of replication, and there's a bunch of those all around, at least in non-linear, in linear DNA in eukaryotes. There's a bunch of origin of replications all over the place, but on an origin of replication, a protein called ORC is going to bind to that. Once ORC binds, it's going to recruit these two other proteins, CDC6 and CDT1. Once these guys are recruited, it's going to recruit another protein called MCM, and these are your helicases. Again, as I don't know if any of you have catched up on 5-1, MCM is your helicase. So during DNA synthesis, it's one of them is going to go to the left, the other one is going to go to the right. It's going to unwind DNA and allow for those replisomes to happen. That's a little bit out of the scope of this exam, but that's what MCM does, essentially. I guess it's worth mentioning that CDC6 and CDT1, they have this ATPase activity. So when they hydrolyze ATP, that's actually what loads the MCM into the DNA. I don't know if that's like extremely relevant, but just so you know, CDC6 and CDT1, they have an ATPase function, which serves to allow MCM to, I guess, be like locked and loaded inside the DNA and ready for it to do the unwinding. Okay, any questions on the pre-RC?
I have a quick question.
Sure.
I'm always a little bit confused by ATPases. So my understanding is that they would take ATP and change it to ADP, correct?
Yes.
So that means that CDC6, is it acting on itself or is it acting on another protein?
That's a good question. I don't really know. I just know that whatever it is that CDC6 and CDT1 do, whatever it is that they hydrolyze, it allows MCM to diffuse inside the DNA. Instead of just like floating above it, it allows it to actually break those hydrogen bonds, get inside into it so that it could fully transverse the DNA, break those hydrogen bonds, and function as a helicase. I don't actually know what CDC6 or CDT is hydrolyzing. I don't know what they're functioning on. Do you have any idea, Rachel, by any chance? No worries. Yeah, so
Sorry.
No,
No, sorry, my laptop just glitched. Like kind of what you said. Are you just confused on like what the ATPase does, or like
I get
like what it
I
functions on.
I get what it does. A lot of the times
Yeah.
it'll say that something is a GTPase or an ATPase, and it never
right?
really specifies exactly what it's acting on, and that usually confuses me.
Oh, gotcha. Okay. Yeah, and these types of like diagrams, it's kind of hard to see because they kind of just look like they're binding to it and like nothing's really happening. I think when you look at the diagrams, you just kind of have to look at what the outcome is and maybe assume like that the hydrolysis of ATP is what led to it. But I don't really know like specifically how to like tell you what it's acting on and what it isn't. It's just kind of based on like the pathway it's in. If that
Okay,
makes
thank
sense.
you.
Yeah, no problem.
Yeah, if it's any consolation, I don't think the ATP function of these two proteins are going to be extremely relevant for your exam. But again, like if you're ever curious, I'm sure Dr. Francis has an answer for you.
No, it's helpful to know that I don't need to worry about that. So thank you.
I personally have never seen a question that asked you about the ATPase function. I don't know if you have, Rachel. I personally haven't.
No, I don't think so.
Yeah. Yeah, I think the main point that Laura just wanted to get out of this was that it's the ATP ACE function that loads the MCM, but I don't really see any foreseeable like questions specifically about like what this is acting on. So hopefully that's a little helpful. Okay, so once your pre-RC is formed, like I mentioned, it's just gonna like sit there. It has to be activated. So before it could go into the pre-IC and start recruiting other proteins, it needs to be activated by CDK. It needs to be phosphorylated. So let's talk a little bit about CDK becoming activated and how it works. So now the cell has established that it has all the nutrients, it's received all the growth factor signals, it's now ready to activate the pre-RC to make it the pre-IC and get cell division going. So how does it transfer into S phase? So I mentioned before that CDK needs cyclin. CDK alone is useless, cyclin alone is useless, they need to be coupled, but they also need to be activated for it to be fully activated. And there's two things that can happen to CDK. It can receive an activating, so CAC, CDK activating kinase, is a protein that adds the activating phosphate. So the CDK cyclin complex alone is useless. It needs to be activated by CAC. I hate that word, but whatever. CAC needs to activate this complex. So once CAC comes along, activates the CDK cyclin complex, the CDK is going to go around, start activating whatever it needs to. But let's say the cell needs to put like a halt. It needs to pause wherever it's at in cell division. We mentioned before that when the cell wants to pause in whatever phase it's in, it's most likely going to inhibit CDK because CDK is the master cell regulator. One way that it could do that is with WEE1. We1 adds an inhibiting phosphate. So when CDK is phosphorylated twice, it's now inhibited. And again, the cell might want to inhibit CDK to allow it to continue growing, to get all the nutrients, whatever it may be, fix cell damage, whatever. Okay, so the cell is paused. It's made sure that it has all its nutrients, all the DNA is fixed. Now what? Now it needs to get rid of this inhibitory phosphate so that it can continue phosphorylating all of the proteins that it needs to continue with the cell cycle. And the protein that gets rid of this inhibitory phosphate is CDC25. So once the cell has decided that it's ready to proceed with the cell cycle, it's going to send CDC25, get rid of this inhibitory phosphate, and now CDK and the cycling complex is just left with its activating phosphate, and it's ready to continue phosphorylating all of the downstream proteins to continue throughout the cell cycle. Okay, any questions so far?
Nope.
Okay. I mentioned before that there was a restriction point or a point of no return. This is where once the cell passes this point, it cannot go back. So once you pass the restriction point, the cell's only choices are completing the cell cycle or going into programmed cell death. So what is the restriction point? Like, what is that characterized as? What that is the release of E2F. So there is this complex, RB and E2F. When this complex is phosphorylated with CDK, as you can see here, RB changes conformation to where it releases E2F. This is what characterizes the point of no return. The point of no return, or the restriction point, is when RB releases E2F and E2F is allowed, it's a transcription factor, and it's allowed to transcribe all of the proteins necessary for the cell cycle. So your point of no return is when E2F is released, is allowed to go into the nucleus, onto the DNA, and start transcribing, and of course, obviously, eventually translating all of the proteins necessary to get the cell cycle going, like MCM, CDT, 1, all of the stuff that we've been mentioning before, like CDC, cyclins, whatever, that's all necessary, or that's what E2F is essentially responsible for. And that's essentially your point of no return. Okay, the pre-IC. So the pre-RC is formed. It has loaded everything it needs to load. The cell has decided it's ready to proceed with the cell cycle. It sends CDK, activated CDK, to phosphorylate the pre-RC. When the pre-RC is phosphorylated, it then starts recruiting more proteins, which is the pre-initiation complex. Yeah, like I mentioned, to get the pre-IC, you do need active CDK to phosphorylate the pre-RC for this full complex to form. Polymerases, yeah, okay, so I don't think we mentioned, yeah, we didn't mention any of the specific proteins that are recruited. I don't think it's going to be like extremely important, but the pre-IC just, again, recruits more proteins, and that's what fires. That's what sends off the helicases in opposite directions and allows the ribosomes to happen. And now you've entered S phase, the synthesis phase. I don't know anything about this.
You want me to take over?
Absolutely, go for it.
Okay, awesome.
So yeah, now we're in S phase.
Can I ask a question?
Yeah.
All right, if we could go back to the last slide. So for creating the pre-IC, my understanding of it was that CDK phosphorylates other proteins and they're added, to the already formed pre-RC complex, and that's what makes the pre-IC. But I believe you said here that the CDK phosphorylates the pre-RC, and that's what causes the other proteins to come. I was wondering if you could clarify that.
Sure. Yes, CDK phosphorylates, or it activates the pre-RC. So if we look at this complex is just kind of sitting here. It's stationary. It's not recruiting anything. It's not until CDK comes, phosphorylates. I don't know exactly what it phosphorylates. It might be ORC, but I'm not entirely sure. To be completely honest, all I know is that it adds a phosphate to something here, and that addition of the phosphate allows the pre-RC to start recruiting more proteins. It's the formation of all of those other proteins post-phosphorylation that creates the pre-IC. We should have included like a picture of the pre-IC because it's a pretty big complex. So I wish I had added a picture of the pre-IC. It'd probably be a little more easier to illustrate and explain. But does that answer your question by any chance?
Yeah, well, I know that the CDK does in S phase, I think it's the very next slide, it does Jesus, yeah. Phosphorylate, it does that to ORC, and I know that that's what makes it so that another PRC doesn't form, and so that transcription only happens once, and so that doesn't happen again during S phase. I guess that's a part of, so the pre-IC is created in S phase then.
The pre-IC is what allows the cell to enter S phase. So the additional, and again, I wish I had a diagram to like illustrate, but those additional proteins that the pre-IC recruits, those are all proteins that are necessary for S phase. Again, I don't know if you guys have looked at 5-1 yet, week 5-1, which is DNA synthesis. Week 5 is not relevant to exam 2, so you don't need to like go hard on it right now. But if you have seen 5-1, in there are several proteins that are involved with S phase. S phase has like these two replicating forks that have a whole orchestra of proteins that are carrying out DNA replication. So those proteins that the pre-IC has recruited, those are all necessary to go into S phase. I don't know if we have a picture of it later on. I don't think we have a picture of the replosome.
I don't think we do.
Yeah, because again, that's also like 5-1 material. So it wouldn't really be super relevant if we had talked about the replosome yet. I really do wish we could have had a picture of the pre-IC, but all of those extra proteins that are recruited in the pre-IC, those are all necessary to go into S phase. So you can't go into S phase unless the pre-IC has gathered its little band of proteins. Does that answer your question, Zev?
It helps. Yeah, I'm still a little confused, but
Okay.
I'll stop by her office hours tomorrow for it.
Sure.
Yeah, she might have like a better answer.
Yeah.
Laura, at least she might, but
Yeah.
yeah.
I don't know if you guys saw her email. She's holding an office hour, I believe, from like 10 a.m. to right before the exam opens. So if you have any last minute questions, of course, you could feel free to attend that. I do recommend it though. Someone may ask this happens all the time when I go to an office hour. Someone asks a question that I didn't even know I had, and I'm like, wow, that question was super important, and I didn't even know I didn't know that. So I definitely recommend going to that. Okay, but anyway.
All right. So yeah, now we're in S phase. Hold on, sorry, my computer's glitching out. Okay, so S phase is where the cell looks for DNA damage. P53 activation can happen at the beginning of this phase as well. And just like a side note, P53 is like a major tumor suppressor. So when it doesn't work, it's P53 in general is found in a lot of like mutations are found in a lot of patients with cancer. So it's really important that it's working and functional, specifically in S phase, because this is where replication is occurring. So if there's a problem or a mutation that slips by, or like a cell becomes overactive, that can lead to diseases like cancer. So it's pretty bad if it's not working properly. So now we have our S CDKs, and they inhibit new pre-RCs from forming because we only want to replicate the DNA once. We don't want more than one replication for DNA. That's not good. There'll be too much DNA for our cells to really handle. So the SCDK levels rise in this phase and inhibit the formation of the pre-RCs by these 3 different steps. So first, they phosphorylate ORC. This prevents the association with CDT1 and CDC6. So they can't bind to ORC, and ORC can't go on and do other things with them. They also phosphorylate CDC6 directly. So once they phosphorylate CDC6, it sends CDC6 for degradation, specifically with ubiquitin-mediated proteolysis. If you remember, that means they're tagged with ubiquitin. Once they're phosphorylated, that means ubiquitin can tag them, and they're sent to the proteasome for degradation. And then when CDC6 isn't available, the MCM complex can't be recruited to join this whole little area for replication to occur. And so because they can't be recruited, the SCDKs can phosphorylate MCM27, which causes them to be exported out of the nucleus. So this whole pre-RC complex can't form, so that DNA only occurs once. Okay, so SCDKs activate the origins of replication to fire so that replication can start. So because this is where replication occurs, so they recruit DNA polymerase and other proteins that are necessary for replication machinery, like replication to occur, because it occurs on either side of the ORC, if you remember. And then once it does this once, that's when it goes and prevents the pre-RC to from forming again, because we only want replication to happen once in S phase. An important part of S phase also is that cohesin rings form around the replicating DNA or the sister chromatids, as in this diagram you can see. The cohesin rings are made of protein, and they keep the DNA strands together throughout replication and into M phase up until anaphase, when they break apart and the sister chromatids need to separate to different sides of the cell. So these cohesin rings are in here for quite a bit to keep them together. This is mostly important to prevent any type of like lack of chromosomes in a certain cell. So like it just prevents the sister chromatids from going off and getting lost or something. So they make sure that each daughter cell gets the right amount of DNA from replication. So this is why they're present in S phase. This is where they come in, and they will stay there through M phase up until anaphase, which is a part of M phase. So once the cohesin rings are around the replicated DNA, there's another checkpoint, which is the end of S phase checkpoint. Basically, the cell makes sure that there's no DNA damage and that the DNA is completely replicated, because if it fails at this checkpoint, then you could result in a ton of different mutations, and we don't want that. All right, so then once we go through those checkpoints, we're now in G2 phase. So G2 phase is once again another growth phase. So the cell grows again and preps for chromosome segregation during M phase. This growth phase is arguably more important than G1 phase, because the cell has already committed to dividing, and it needs to make sure that there's enough resources for the cell to divide, because the cell's basically at this point going to divide no matter what. So basically, it needs to make sure that the M phase cyclins are not activated too early, because if they are, then the cell wouldn't have grown enough for division, and you could have some problems with division if the cell's not big enough with enough like machinery or proteins around for division to occur. So once again, we have this like inhibitory pathway with cyclin and the like WEE1 and CAC and all that. So during G2 phase, the MCDK levels start rising, but they're inactive. They're inactive because WEE1 has phosphorylated the inactive site on MCDK, and CAC is also activating, phosphorylating the active site for MCDK. So basically, you have these two phosphates in two different spots. You have one inhibiting it, one activating it. So just they cancel each other out. And that does this until the cell has grown big enough for M phase and for cell division to occur. So once that happens, then that is when CDC20 will come in and take off the WEE1 phosphate, so that it's no longer inhibiting the complex, and then it can go off and do its thing. So once we're big enough to divide, that's when the M cyclin levels now become active. We need to have enough resources. And once again, we check for any DNA damage that might have slipped through the S checkpoint. Do you guys have any questions on G2 before G2 phase is kind of the later one, but do you have any questions so far? No, cool. So late G2 phase, this is where we proceed with M phase when the MCDKs become active. So we're not in M phase yet. This is when the CDC25 takes the phosphate. I think I said CDC20 before. I meant CDC25. CDC25 takes the Wee1 phosphate off of the inhibitory site on the MCDK complex. So now our MCDK is active. Our levels were already rising, but this positive feedback loop allows for more MCDK to become active very, very quickly. It's like almost exponentially, like the graph goes up very quickly. So basically what happens is this CDC25 in the first scenario is only partially active. We don't actually know how the original activation begins, but it's not fully activated. So it removes the inhibitory phosphate, and then by removing that phosphate, now the active MCDK can phosphorylate the CDC25 that was only partially active. And now it becomes fully active. CDC25 can remove more phosphates. With more phosphates being removed, we have more active MCDK, and it just does this loop. So MCDK phosphorylates CDC25 to make it active to remove the inhibitory phosphate from other inactive MCDKs. And this is what allows us to get into end phase. You guys have any questions on G2 before we start end phase? Or the positive feedback loops? She might ask you about those, but if you have any questions, we can keep going. So this is just quick M phase overview. You have prophase, prometaphase, metaphase, anaphase, and telophase and cytokinesis, which are usually clumped together. So in prophase, we still have that active MCDK. An important part of prophase is the breakdown of the nuclear envelope and the condensing of chromosomes in the nucleus. Well, the nuclear envelope breaks down, so I guess they're not really in the nucleus anymore, they're just in the cell. So that's prophase. Prometaphase is when the microtubules grow and shrink towards their respective locations. This is that push-pull mechanism that we'll talk about in a little bit. And they attach to the kinetochores on the sister chromatids. Metaphase is basically when they just line up on the metaphase plate, which is just the middle plate. Anaphase is when they separate, and telophase and cytokinesis is when the cells divide and everything kind of decondenses and the nuclear envelope reforms so that we have our two daughter cells. That's just a quick overview of M phase. So now I'll go a little bit more into detail. So prophase is when the active MCDKs, I think, are the most active in M phase, is during prophase. Basically what they do is they phosphorylate these proteins, one called lamins and the others are condensins. So when they phosphorylate lamins, basically they just break down the nuclear envelope so that we can have access to the DNA inside of it. And then to condense our chromosomes, the MCDKs phosphorylate condensin, and they become more like sausages rather than spaghetti. They become super dense. And then they also phosphorylate various MAPs and motor proteins to help form the spindle. So that's prophase. So you have those three sets of phosphorylation. So sequential phosphorylation is just the idea that phosphorylation needs to happen in a specific order. An example was in the survival signaling pathway with AKT, protein kinase 1 and protein kinase 2. That's just an example. So basically what happens is the CDK, so in this case, talking about MCDK, needs to phosphorylate a substrate before the actual enzyme can also phosphorylate the substrate. So in this specific example, the CDK phosphorylates the substrate and creates a binding site for PLK1. So PLK1 on the PBD domain will bind to the phosphate put there by CDK. And then once it binds to the phosphate that CDK put on the substrate, the PLK1 enzyme is now active and then can use its kinase domain to phosphorylate the substrate at a different position. This is just an example of how the CDKs are called the master cell cycle regulators, because if CDK didn't phosphorylate the substrate, then PLK1 can't do anything. So they're pretty regulatory in what they do. And without them, you wouldn't be able to do a lot of different things in the cell cycle.
Okay, so I realized we may have glossed over the microtubule section, but we could talk about that now. I don't mind taking it if that's okay. Okay, so microtubules, you have a few different amount of them. The main one that we're going to be talking about, we're obviously going to be talking about microtubules, but just so you're familiar with the cytoskeleton, you have your actin, intermediate filaments. These are more relevant with muscle contraction, like calcium diffusion, things that affect, not super relevant to the scope of what we're going to be talking about in this course. Like I said, we're mainly going to be talking about microtubules. Not that these aren't important. I do recommend that you do get a little more familiarity with these, but again, like these are more involved with like muscle contractions. The ones that we are going to spend more time on are microtubules, because these are essentially like your roads inside of the cell. They're also the reason that there's like a microtubule interlude right in the middle of what Rachel was talking about in cell division. It's not random. I promise. It's microtubules do play a really important role in chromosome segregation, so that's why we've interluded into this segment right now. And like I mentioned, the main one we're going to be talking about are microtubules. Okay, so microtubules, it's a little hard to see here, but essentially what microtubules are, it's this tube-like structure that's composed of this dimer, these tubulin dimers. So you have your tubulin alpha, your tubulin beta, and like I said, these are dimers, so they go together. And it looks like this on a very microscopic scale. It looks something like this. So your alpha end, it has a GTP binding pocket, but it's inside. Whereas the beta tubulin, or excuse me, the beta dimer has a GTP binding pocket that's exposed. And again, I'll show you another picture to clearly illustrate this. The minus end is usually associated with the alpha, because again, the way I see it is the plus end of the dimer is, it's plus because this is where it's going to polymerize. This is mainly where the microtubule structure is going to grow. So again, I'll show you another illustration that better captures this, but alpha is the minus end. There's not going to be a whole lot of polymerizing on this side, but on the beta end, on the plus end, this is where more microtubule dimers are going to be accumulating. So that's how I remember that the beta end is like the plus end, or that's how I view the plus end. Plus and minus, just so you know, these are not charges. There's no like electrical differences between the two. The plus is just to tell you this is where it's polymerizing. So, okay, so in the structure of microtubule, tubulin are heterodimers, meaning that they're composed of two different dimers, alpha and beta, like I just talked about. These are microtubules are hollow tubes. So as you can see here, there's a lumen, this like empty hollow space. Okay, so I'll talk a little bit about like the tubulin structure. As you can see here, this is the dark green is the beta, where the light green is the alpha end. Notice that when the microtubule structure is growing, it is the beta dimer that is exposed. And that's why this is why I see it as like the plus end, or this is how I remember that polymerization happens at the plus end, because on the beta dimer, this is where more microtubule dimers are going to continue accumulating and continue polymerizing the microtubule structure. The minus end, however, is what connects to the microtubule organization center, which I'll talk about in a second. Oh, which I'll talk about right now. So what starts the microtubule from forming in the first place, all microtubules start from this microtubule nucleation section, which is oftentimes called, it's the centrosome. So all of the microtubules are going to originate from the centrosome. Inside here, those little sausages, those are called centrioles. I'm pretty sure those are vestigial organelles. So our cells don't really use those anymore. They're just kind of there vestigially. They don't really serve a function anymore. They don't really do anything. But what does do things is the centrioles. So, the centrosome. The centrosome is like this globe thing. This is where the microtubules are going to be polymerizing from. This is their start point. Again, the centrioles, those sausages inside the globe, I don't think they really do anything. I'm pretty sure they're vestigial, but it is the globe that does do things. So the beginning or at the surface of this globe where microtubules start polymerizing. That's called the gamma tubulin. So this is like a close-up of what the gamma tubulin looks like. And what happens is you have your microtubule dimers, the alpha and the beta. They always come together. You're never really going to see these separate. In all my years of life, I've never seen these like separate. So you don't have to worry about like what happens if the alpha and the beta dissociate. It is possible for the dimer itself, for the pair itself to dissociate. That's we'll talk a lot about that in a second. But you're never really going to see this dimer like falling apart. The beta and the alpha coming falling apart from each other. That doesn't really, or I'm sure it can happen, but it's not really is something that you have to worry about. Anyway, the alpha end is what's going to connect to the gamma tubulin, which again, this is just a close-up of the surface of this globe over here. So these red rings that you see on this globe, that is just this. This is the starting point. The tubulin dimers are going to find the centrosome, and then they're going to continue polymerizing by the beta end. So it's going to polymerize by the plus end, which is the beta dimer. Any questions so far about any of this? Okay. Dynamic instability. The main idea of this is that microtubule structures are constantly polymerizing, they're constantly growing, and they're constantly falling apart. So that's the main idea of dynamic instability. And how these structures form and fall apart all depends on their GTP hydrolysis. So notice here in this microtubule structure, you could see these red dots. These red dots just represent GTP. So when the tubulin is connected to GTP, it is able to polymerize. So for instance, if you look at this one, notice that there are no red dots. There are no GTP on any of these tubulin dimers, which means that this specific microtubule cannot continue polymerizing. It cannot continue polymerizing because it is not bound to GTP. Unlike in this one, there's plenty of red dots. There's plenty of GTP. So the tubulin dimers are allowed to continue polymerizing. And that's what it's doing right here. As you can see, this is growing into this and is growing into this. But as you also noticed, there's some hydrolysis happening. Let me just make, okay, yeah. There's hydrolysis happening. So the tubulin dimers are capable of GTP hydrolysis. And that's, as you see here, there used to be red dots here. There's no longer red dots. Those red dots represent just GTP. The reason that you don't see GTP over here in this one, it's because it's already hydrolyzed. So in this little analogy, I like this. If the rate of GTP hydrolysis is greater than tubulin addition, so if you get to this point, if you get to the point where GTP has hydrolyzed everything before more tubulin dimers are added, your microtubule structure is going to shrink, it's going to depolymerize, it's going to fall apart because it's no longer stable. However, if your rate of tubulin addition is greater than GTP hydrolysis, like you see in this scenario, you're adding GTP dimers faster than it is able to hydrolyze. And if you have this scenario, then you're going to grow, you're going to polymerize your microtubule, and you have essentially what's called the GTP cap. Once you lose the GTP cap, or once all of these GTP get hydrolyzed into GDP, then you have this situation when your microtubule structure is completely GDP bound and there's no tubulin being added, it's going to start falling apart like you see here. Okay, any questions on dynamic instability? Okay, so there's some drugs that can affect microtubule assembly, and Laura loves these. So a very popular question that I anticipate for you guys is you might see several scenarios that's like, draw a graph that illustrates microtubule assembly if you add a bunch of Taxol or if you add a bunch of colchicine. So just like play around with questions like that in your mind. So there are some drugs that affect microtubule assembly. Taxol stabilizes microtubules. So if you are consuming Taxol, not that it's like, normal, but if you are taking Taxol for whatever reason, it prevents your cells from depolymerizing. So it prevents it from hydrolyzing that GTP. Whereas colchicine does the opposite, is a polymerization inhibitor. So it prevents your microtubules from growing. Vinblastine, also a polymerization inhibitor. So yeah, just be vaguely familiar with what these does. Taxols allow the polymerization to occur, whereas colchicine allows depolymerization to occur, or it inhibits microtubule formation. Okay, so any questions about these drugs? Oops. Okay, yeah, so Laura loves questions like these, so definitely just anticipate some questions about microtubule assembly. She might ask you to draw a graph. I've seen that in, I think, like every exam that I've ever taken with her.
Yeah, she usually has some type of graph. You should draw.
Yeah, so just get a little familiar with that. You guys have probably done it in your problem sets or something like that, so I'm sure you're probably familiar with that.
Would that be the graph of like the different phases of microtubule formation?
Yeah. Okay. Yeah. So a question that I've seen before is like, what if there's just like a bunch of colchicine before anything happens? Draw a graph that illustrates microtubule assembly if before assembly has started, the cell is already exposed to a bunch of colchicine. And a graph like that, obviously I can't like illustrate it right here, but what that graph would look like essentially is you'll have a very long lagging phase. So you guys probably remember in that lecture, you have a lagging phase. That lagging phase is just when the microtubules are assembling. So there's a lagging phase. It takes a little bit for the dimers to find the centrosome, to start connecting to the gamma tubulin. It takes a little bit for that to happen. So that's why you have a lagging phase. But once this has polymerized, once there are enough tubulin dimers on your gamma tubulin, then you have like an exponential phase. Then you'll notice that graph kind of skyrockets. It skyrockets because polymerization happens very quickly after that lagging phase is completed. If your cell is exposed to a bunch of colchicine, if your cell is exposed to a bunch of colchicine before you've reached the polymerization phase, it's going to take a very long time for this polymerization to happen. So you could expect a very long lagging phase. So that's just some kind of questions you should play around with. Or the opposite, what if your cell is exposed to a bunch of Taxol right at the very beginning? In that case, you'll have a very short lagging phase and your polymerization will just kind of skyrocket from the beginning. So those are just some things you can anticipate. Sorry, did I answer your question? By any chance?
Yeah.
Okay, word.
Yeah, thank you.
Yeah, no
Fun
worries.
fact, I take colchicine.
No way.
Yeah, not for gout, for an autoimmune disorder, because it does the same thing. It's actually pretty cool. They're looking at colchicine because apparently it could possibly help COVID patients. So
Oh, I had no
pretty
idea.
cool. I know, right?
I almost said congrats. I don't know if that's something actually, but
But there is something called colchicine toxicity, which is when you have too much inhibition of microtubules. So
I see.
there's such a thing.
Cool. I didn't know that. All right. MAPS, microtubule associated proteins. Okay, so there's this protein called tau. It's mainly expressed in the central nervous system because tau, really what tau does is it stabilizes neurons. So obviously here you have a neural cell. What tau does is just it kind of aligns itself along the oh god, I just took a neuro class along the axon. It aligns itself on the axon and it stabilizes it. However, in some neurodegenerative diseases, or if your tau is dysfunctional, like in Alzheimer's, for instance, your tau protein can be really messed up. This isn't the case where you have too much, right? Hyperphosphorylation no longer bonds
Yeah.
the aggregate. Okay, yeah. So in the instance where you have too much tau. Okay, so let me just recap. Along your axon, you have microtubules that's stabilizing your axon, keeping it healthy. Tau is like this little orange spike thing that you see here. Tau is just stabilizing the microtubules that's lined up along your axon. In Alzheimer's disease, for instance, you have a hyperphosphorylated tau where there is an abundance of tau, and instead of tau stabilizing your microtubule structure on the axon, it's actually aggregating within itself. Protein aggregation is obviously super bad because it disrupts the function of the protein. If proteins just aggregate together and clump together, obviously there's no room for them to function properly. So if tau is aggregating with itself and it's not functioning with the microtubule structure, then your microtubule is going to start dissociating. And obviously, if your microtubule dissociates, that's going to have really bad, that's going to be a pretty bad outcome on your neurons. And that's essentially the premise of neurodegenerative disorders like Alzheimer's disease. Not for every single one of them, of course, but specifically for Alzheimer's. Okay, any questions on MAPS or tau? Okay. Motor proteins. Okay, so why are we even talking about microtubules? Well, we're shortly going to jump back into cell division. That's how we got into talking about microtubules. But moreover, on microtubule structures, before I called them like the roads of the cell. They're considered roads because they really are like roads. You have these two proteins, these motor proteins, kinesins and dynesins. These are essentially, they're motor proteins that transport vesicles. These vesicles could contain anything, and they transport them all around the cell. So the way that these motor proteins work, okay, here's where I want to make like a really important differentiation. So microtubule assembly polymerizes with GTP. So GTP is specific to microtubules and microtubules only. Motor proteins, these guys function with ATP. ATP and GTP cannot be used interchangeably. I saw like a few people, like in the fall semester, I saw a few people who kind of use them interchangeably. Please do not say that. Motor proteins use ATP, not GTP. So the way that these guys work, and there's a bunch of proteins, there's a bunch of movies that kind of illustrate this for you. If you go on the movies folder on the Moodle, you can see a bunch of cool videos that show you what they do. And like I said, what motor proteins like kinesins, dynesins do is they just transport vesicles. Kinesins walk towards the plus end, or dynesins walk towards the minus end. And the way that they work, I mentioned before that they work with ATP hydrolysis. And the way that it works is it is this cyclic motion of ATP hydrolysis and ATP addition that allows these legs to swing forward. So if I remember correctly, when ATP is hydrolyzed into ADP, this globular head is going to swing forward. Does that sound right, Rachel? Okay, so
yeah, it swings forward.
Okay, when it hydrolyzes, it swings forward. That allows for ATP to diffuse in, which locks it in place. And then again, that cycle is just going to continue. So it's the cyclic motion of gaining ATP, hydrolyzing ATP that allows these globular heads to swing back and forth. And again, if you want like a visual representation, there's a bunch of cool movies in the movies folder on Moodle that you could check this out. Okay, there's a whole family of kinesins. I don't know if there's a whole family of dynesins. I think, yeah, I don't remember. I almost said Rachel. I don't remember talking too much about family of dynesins, but there's certainly a family of kinesins. You should be familiar with some of these only because I have before. I can't promise you that this will come up in this exam tomorrow, but I have seen questions related to some different kinds of microtubule kinesins.
Well, yeah, because one of them, it's in it. I think it's kinesin 13. One of them goes towards the negative end.
Oh, really?
Yeah, I know one of them does. I don't remember
Oh.
which one.
Yeah, it looks like here. Oh wait.
Yeah, I one of them, you should just be familiar with which way they go, because some of them go, like a majority of kinesins go toward the positive end, but some of them do go towards the negative end or both. So just make sure that you know like which ones do what.
Yeah. Yeah. Okay, so here's your family of kinesins. Like Rachel said, definitely familiarize yourself with which ones go in which direction. Generally, kinesins go towards the plus end, whereas dynesins go towards the minus end. But there are some kinesins that can go in both directions. Me and my best friend Jason, we're having this argument where we want to isolate our kinesins because I think my kinesins are faster than his. So I want
No.
to race our kinesins. Yeah, we actually asked Laura. We were like, Laura, how do you
No.
isolate kinesins? Just like hypothetically. And she was
Not
like,
Jason.
yeah, we're gonna race our kinesins one day, and I
God.
promise you, mines are faster than his. But anyway.
Are you just saying yours are faster because he's in a wheelchair?
Oh no! Well, actually, he was like, "Your kinesins are slow because you know they walk slow." I was like, "At least they can walk."
Oh, stop it! You did not.
It was bad. It was super bad. But
Oh my God!
I just
No,
swear,
it's okay, guys. No, Jason makes fun of himself. It's okay. It's okay.
Jason's my lover, but
No, we love Jason. He does it himself. We just a side note, just to make you guys laugh for a quick second. You know, at Mullins, I don't know if you guys have been, but for COVID testing, there's a sign that says "Stand here," and Jason took a picture of it. He's like, that'd be great, but I can't stand. And I'm like, oh my God,
this
Jason.
man can't stop me because I can't stand.
Yes, exactly. I'm like, oh God.
Yeah.
Anyways.
Point of the slide is that my kinesins are very fast. Back to the
Yes.
cell cycle.
All right, so this is me. All right, so spindle formation. Before we do that, there's certain types of microtubules that have different jobs. There's asters, kinetochores, and interpolars. Don't get kinetochore microtubules mixed up with kinetochore proteins, which we'll talk about in like the next 2 slides or something. They're different things. Kinetochore microtubules grow from the centrosome and attach to the kinetochore on the sister chromatids. So when you're talking about kinetochore microtubules, just make sure you mention that it's microtubules and not the actual kinetochore, because that's what it attaches to. Aster microtubules grow from the centrosome and attach to the cell cortex. So they're like on the outermost sides, like closest to the cell membrane. They don't really go that far into the middle. And then interpolar microtubules grow from the centrosome and overlap in the middle. If you see that like pink line with the with the black like dots in it, those are proteins that hold them together. And this is where they kind of make that plate, metaphase plate. This is like the line where they are. And they can grow, the interpolar microtubules grow and shrink. They're responsible for the separating of the cell when we do like the push and pull mechanism. So that's what those ones are. So those are the 3 types of microtubules. They each do different things. And the centrosome is that little green, the spindle pole is basically with the centrosome. They're the same thing. So this is that push-pull thing I was talking about. So the main idea is that spindle formation requires an array of opposing forces. So we have kinesins and dyneins, which we just talked about. So kinesins and dyneins, they walk towards opposite ends of the microtubule to create opposing forces. So the dyneins walk towards the minus end and they create a pull, whereas the kinesins in this specific example, or in this specific opposing force for spindle formation, we use EG5, which is a kinesin. So it walks towards the plus end and it creates the push mechanism. So you have this push-pull because they're both walking in different directions, specifically on the interpolar microtubules that overlap in the middle. And that's basically what they do. They walk towards the different directions. And if you have something, so like I think it's monoestrol, it's an EG5 inhibitor. And basically when you inhibit EG5, the spindle kind of collapses because you would only have the dynein motor proteins going towards the negative end. And they would just be pulling the spindle. They wouldn't be pushing it also. So as a result, the spindle would just collapse. So you need both of these opposing forces to create the actual spindle. Otherwise, the spindle will most likely collapse. So the same forces also, just to add on, that work here also work in anaphase when we pull the sister chromatids apart. So like I mentioned before, the kinetochore is located on the centromere region of the sister chromatids. This is on a replicated chromosome. So the kinetochore microtubules attach to the kinetochore, and this is just where they attach and they pull the sister chromatids apart from that location. The kinetochore is made up of a bunch of different proteins. The actual like diagram of a kinetochore is actually pretty complex. But the specific proteins that you should know that make up a kinetochore are MAD, BUB, and CDC20. Yeah, that's basically it. The kinetochore microtubules attached to them, and that's where they get pulled apart. So metaphase is where the sister chromatids line up in the center, or the metaphase plate, whatever you want to call it. Basically, what happens here is the cohesin rings are still holding the sister chromatids together, help them align along the plate, and that's really it. They just line up on the plate. The real action happens in anaphase. So yeah, so this metaphase to anaphase is like the spindle. This is the spindle assembly checkpoint. So this is the checkpoint that if we pass, we're now in anaphase. So basically, what happens is it makes sure that all the sister chromatids are aligned and that the spindles are all attached properly at the kinetochore so that we can pull apart the right sister chromatids. Because if we mess this up, we could end up with something called like, oh my God, what's it called? Is it just trisomy, right? With
non-disjunction.
Non-disjunction, yeah, that's it. Where we could end up with the sister chromatids don't separate. And we get one cell that has too many chromosomes and one cell that doesn't have enough. It's something that leads to Down syndrome with trisomy. So basically what happens is the cell will inhibit chromosome segregation unless there are proper microtubule attachments at the kinetochore. And then this MAD-BUB CDC20 like complex is on the kinetochore. And these are what sense, or the MAD and BUB sense that the microtubule is attached properly. And they sense the tension in it. So when the microtubules start to pull the sister chromatids apart, they sense this tension to make sure that there's an equal tension on both sides of the kinetochores, on either sides of the chromosome, because you want the same amount of tension pulling them apart. Otherwise, you will end up with something like non-disjunction where they don't separate properly. And if MAD and BUB are like, okay, yeah, there's equal amounts of tension on either side, then it will release CDC20.
How do you know all this?
Yeah, and then CDC20 goes on to bind to something called APC, and it creates this CDC20 APC complex. So when it binds to APC, it makes APC active. And then what APC does is it targets this protein called securin, and securin is basically just inhibiting this other enzyme called separase, and it inhibits separase because we want to keep the cohesin rings on the sister chromatids up until this point. So securin is doing its job up until CDC20 and APC target securin, and it basically targets it for degradation. So it gets ubiquitination and degradation of securin occurs, and it's sent to the proteasome for degradation. That's that brown protein attached to the blue one. And then, so now that securin's degraded, separase can go on and what does it do? Cleave cohesin rings. So it cleaves the cohesin rings that are keeping the sister chromatids together. And once they're separate, or once they're cleaved, the sister chromatids can now be pulled apart in anaphase. So this is the specific point in which we're now moving from metaphase to anaphase, because once those cohesin rings are cut by separase, the sister chromatids are going to pull apart because there's already tension in the microtubules. They're just being held together from those rings. This is also the point where the M cyclin CDK activity drops significantly, specifically at the beginning of anaphase, because we don't want we don't want the phosphorylation of other proteins that MCDK is currently doing. So we want that to drop off at the beginning of anaphase. So this is the point where MCDK cycles drop significantly. And now we're in anaphase. So there's two types of anaphases, whatever. There's A and B. Some organisms like prokaryotes, eukaryotes do one and then the other. Some do them at the same time, but it doesn't really matter. So in anaphase A, what happens is the chromosomes are pulled poleward. So they're pulled to either poles that based on the forces generated by the kinetochores. The kinetochore microtubules shorten. So if you look at the diagram, the kinetochore microtubules are what are attached to that little red dot on the chromosomes, and those just shorten significantly. And they just pull the sister chromatids to the poles, like either poles. And you'll notice that the spindle poles stay in place. The spindle isn't growing or shrinking at all. The only thing happening really is that the kinetochore microtubules are shortening. Anaphase B is different. So anaphase B, the spindle is actually moving, or the centriole, yeah. So what happens is the poles are pushed and pulled apart, similar to how the spindle is assembled. So the sliding force between the interpolar microtubules in the middle is coming from opposite poles. So the pulling force acts directly on the poles to pull them apart. So the microtubules will grow at the plus end. So if you look at the difference, if you look at the first part of anaphase B, you'll see that the first step is that the sliding force is generated between the interpolar microtubules to push and pull them apart. And then as a result, the spindle poles will be pulled apart in opposite directions. So the entire spindle is growing. It's just getting longer. And that's what eventually will pull the sister chromatids apart. So those are the main two differences between anaphase A and B. You just need to know that that there is a difference and that this is what they are. But do you guys have any questions before we get to cytokinesis and telophase? No? Cool. So this is the last step, arguably the easiest step. At this point, the sister chromatids have reached the opposite poles. There's a cleavage furrow, which is in this that little dip in between the two. Yeah, right there. It's created by this contractile ring that's made of actin and myosin. And it gradually just deepens until it hits the spindle and then eventually splits. So it just gets more narrow until eventually the cells split thanks to this contractile ring. And now that the M-cyclin CDK activity is dropped, the spindle will break down. The nuclear envelope will reform, because if you remember at the beginning of prophase, we needed the M-cyclin CDK to get rid of the nuclear envelope. So now that the activity is significantly dropped, the nuclear envelope can reform around the DNA. The chromosomes can uncoil to go from sausages to spaghetti, because that was another thing that M-cyclin CDK did in prophase. It made them from spaghetti to sausages. So once again, the activity has dropped, so the chromosomes can go back to their original resting state, which is in long, strands of spaghetti, I guess. And because this allows for use in metabolic processes. So that's something she likes to ask about, is the certain levels of M-cyclin CDK. So just remember that in prophase, the levels increase very quickly through that positive feedback loop. And it allows for the nuclear envelope to break down and for chromosomes to condense. And then when we get to anaphase, they drop significantly, so that in telophase, the nuclear envelope can reform and the chromosomes can decondense. So here's just some cell cycle main ideas. These are a lot of the main ideas she likes to ask about. So the cell cycle events happen in a particular order. You need to go in this order for the cell cycle to function properly. This is why we have our checkpoints, so that we don't skip steps and don't go too quickly or too slowly. The cell cycle events involve cyclical activation or deactivation of proteins. This is often by the means of phosphorylation with fresh ATP, like we talked about with kinases. CDK must be bound to cyclin in order to be active. If it's not bound to cyclin, it doesn't do anything. CDK is also the CEO of the cell cycle because it regulates particular proteins in different phases, like we talked about with sequential phosphorylation. Different phases of the cell cycle have their own cyclins, like S-cyclin, M-cyclin, And then each cyclin is created and degraded in a particular part of the cell cycle, like we talked about with M cyclin. We needed it in prophase, and then we don't need it in anaphase or telophase. And number 5, regulation for proper functioning requires activation and deactivation as well as inhibition. So they're not positive or negative, it's just whatever the cell needs at the time. So if we need to inhibit replication, we will, and if we need to enhance it, we will. And that's it.
I don't like that this 4 is bigger than the other numbers.
Yeah, I don't either.
Whatever.
Puppies!
All right. All right.
You guys are going to do great.
How are you guys feeling so far?
That's it.
All right.
Decent, yeah.
Okay. Okay.
Cool.
I mean, I feel prepared enough to be able to rewatch a lecture and answer the question.
Yeah,
Good.
same.
So
Good.
I
Awesome.
don't
That's-
think I could
You
do
have
it.
24 hours to do all of that.
Well, I definitely recommend hitting up Laura at her office hour tomorrow morning. You know, maybe if you don't have like any specific question in mind, she might just talk about something else on her own perdition, and that might you know, it might raise some questions that you didn't know you even had. So I definitely recommend checking that out tomorrow morning if you can.
Yeah. She'll, yeah, usually she's good at answering questions.
So I meant to say her own prerogative, not her own perdition. Like that.
All right. Well, thank you.
You're welcome. Have a good rest of your night, guys.
Thank you.
See you guys. Are they gone?
The recording has stopped.